not call conspiracy everything this people calls a conspiracy. Do not fear what they fear, and do not dread it.
Do you feel it in the air? Everything is strange in a world wasted by fear.
You hear people make comparisons all the time about how your brain is like one big, ultra-sophisticated computer, and that's fine. Recently, scientists working with mice have been able to send new signals into their brains, ones that seemed so natural, the mice may have no idea they weren't their own. It seems a little sci-fi, but it's real research. Studies center around a branch of neuroscientific research called optogenetics, which makes use of light-sensitive proteins inserted into neurons through genetic manipulation. Once the proteins are there, researchers can control the firing of those neurons using just a flash of light. With it, we've been able to get groups of neurons to fire and even induce behavioral changes in rodents. Like one experiment used optogenetics to get rats to walk in circles, which is pretty amazing. Also a little upsetting.
Why is it that I have to do this now? This is my life.
To advances in tech and biology, a group of scientists recently figured out a way to target specific individual neurons rather than a relatively large patch of tissue. They published their results in April 2018 in the journal Nature. And brace yourself for some sci-fi, because they're doing it using holograms. Visual holograms, the kind you see in Star Trek or Star Wars, use multiple beams of light to project an image. But holography in general is really just about light interference, and that's what this technique relies on. The team called their method 3D SHOT, which stands for three-dimensional scanless holographic optogenetics with temporal focusing, which I'm glad they have such a good acronym for that. And because that firing appears to really closely mimic the way their brains would naturally detect and process stimuli, it's likely that to those mice, any experiences induced using 3D SHOT would be pretty realistic. I mean, unfortunately, we cannot ask the mice about this, but if we can stimulate their brains into firing in the same patterns as it would when, say, seeing, feeling, or smelling something real, that seems reasonable. One way we can test this hypothesis is by observing actual behavioral changes, like a flick of a switch causing a mouse to stop in its tracks. If scientists are able to make neurons fire realistically, though, and if those experiences feel real even though they're not, well, in theory, this technology could eventually be used to alter, like, memories by editing the way neurons associated with those events fire. We could make mice perceive things that aren't really there, all with the flash of a few lights. The research team responsible for 3D SHOT already have a few ideas about how they're going to sort of copy and paste brain activity to achieve something like this. If they can measure the sequences of activations in specific neurons involved in the behavior they want to program, 3D SHOT could feasibly send that pattern into a different brain. Basically, programming it. Brain programming.
The video was recorded in China and was published by People's Daily on Twitter. It shows a herd of sheep walking in circles for more than 12 days. After being uploaded to Twitter, the video was quickly shared to many other social media and internet users immediately began. The thing is, experts were baffled and had almost no answer to why this was happening. Matt Bell, a professor, stated that it could be because the sheep are tired of being in the pen for long periods, and that leads to a stereotypic behavior such as this one. That would maybe explain this particular video. However, a few days later, many other videos started being uploaded to YouTube, TikTok, Facebook of similar things going on, but this time all over the world with different kinds of animals.
Salt pans, an example of extreme living conditions. The high salt content is deadly for most living creatures. With one exception, halobacteria. This is where they really start growing, and that makes them interesting to scientists. In 1973, Dieter Oesterheld and Walter Stockenius discovered a protein in the membrane of these halobacteria whose activity is controlled by light. It permits certain ions to pass from one side of the membrane to the other. The scientists called it bacteriorhodopsin. Other bacterial proteins activated by light, such as halorhodopsin, emerged as well. The researchers still had no idea how valuable this discovery would one day be. "30 years later, this new science called optogenetics is investigating how to transfer light-activated proteins into nerve cells and then control them using only light cells. Optogenetics links gene technology with optical methods. Can it help us to influence neuronal networks and one day even overcome diseases like Parkinson's, epilepsy, or blindness? The Max Planck Institute for Biophysics in Frankfurt. This is where the optophysicist Professor Ernst Bamberg works. Light for him is a very special tool. With the right stimulus, he can use it to open doors. He aims to use light to enable nerve cells to open membrane channels, keeping his team on the move as he does so.
The procedure is really very simple, and that is what makes the method so popular. We can quite easily use molecular biology to implant light-sensitive proteins into cells, preferably cells that can be stimulated by electricity. In other words, nerve cells on the one hand and muscle cells on the other. And then the cells can be activated or deactivated by using light.
In spite of the discovery in the 1970s, the starting signal for optogenetics came only in 2002. Max Planck research scientists, including Ernst Bamberg, found another membrane protein that reacts to light in the green alga Chlamydomonas reinhardtii. It permits the alga to use light for orientation. With the molecular biological tools now available, the researchers succeeded in transferring the gene for algae rhodopsin to frogs' eggs. These now produce the corresponding protein. It was a surprise to find that it is a light receptor, that is, rhodopsin, and an ion channel combined. So it was given the name channel rhodopsin. The remarkable thing is that the channel reacts to blue light. It opens up and lets positively charged ions flow into the cell. Optogenetic scientists use this mechanism by smuggling the gene with the building instructions for the channel rhodopsin into the genetic material of the nerve cells, so that this channel can then work inside the membrane. If it is stimulated by light, positively charged ions flow into the cell. This changes the tension across the membrane, and the neighboring original channels also open up. In this way, the cell is depolarized and an action potential arises. But the opposite effect also occurs. Halorhodopsin, which comes from halobacteria, reacts to orange light and smuggles negative ions into the cell. This then deactivates the cell, switching cells on and off using light, a fascinating prospect.
This is a method which is less invasive, as we say. In other words, it means I no longer need electrodes, which I normally always have to use in neurobiology. Of course, the implications are enormous. I can switch light on and off. I can make it work exactly as I like. I can even stimulate individual parts of the cells separately without too much difficulty, and then can check on the signal.
First of all, we breed them in bacterial cultures. To transport the genes, the scientists use viruses which they have reprogrammed. The advantage is that viruses are very efficient at transporting the information into the required cells. Finally, light-controlled proteins are a good example of how basic research can open up completely new fields. Who would have thought that the membrane proteins of tiny single-cell organisms could play such an important role?
Smart dust.
This powder-sized chip, and that's a salt crystal, so this is a small thing. is something called the Mu chip from Hitachi. It's the smallest commercially available RFID system in the world and can be pulse powered by radio waves. It doesn't require a battery. You can literally scatter this stuff like dust or embed it into a sheet of paper. And you know what the really interesting thing about this technology is? This was commercially released 10 years ago. So the inevitability of smart dust. So what is smart dust? Well, smart dust, of course, isn't a new concept. It originated with DARPA back in the 90s. And it's general purpose computing, sensors, wireless networking, all bundled up into millimeter scale sensor modes, drifting in the air currents, flecks of computing power settling on your skin, ingested, monitoring you inside and out.
But in the future, every square inch of every city will be alive with intelligence. Because every street and every building will have a network of microcomputers built right into them. Dr. Chris Piester calls it smart dust.
A smart dust particle, or mote, is a wireless sensor with sensing, computation, communication, and power in one package.
These all-in-one microcomputers will be small, very small.
The size of a mote today is about the size of a grain of rice. And we've shown that we can make the circuitry small enough and light enough that eventually it will be possible to make things that are on a sub-millimeter size scale.
Tiny specks of computer smart dust will form a vast invisible network that can help manage the infrastructure of even the largest city.
Smart cities in the future will take this low-power, inexpensive, small technology and basically distribute it everywhere.
No matter how we live in the future city, it will be radically different.
New research in Britain has raised concerns about the potentially harmful effect of nanoparticles. The emerging technology is used in a variety of medicines and even sunscreen. The research found that the small particles could damage the DNA of cells. They're tiny microscopic particles which can store and release drugs in specific areas of the body. As a result, nanoparticles are increasingly being used in medicine and surgery. But because the particles are so small, there's been growing concerns about potential toxicity.
There's an emerging sort of body of evidence that says that for some particles of certain scales and of certain materials, that there are potential health risks.
New British research has added to that concern. It found nanoparticles could damage cells without even entering them. Scientists found the particles change the function of cells and how they communicate with other cells.
What this study shows is that even with a barrier between the blood supply where the nanoparticles are and cells, that there can be damage to cells, and not just a bit of damage, but significant damage to the DNA.
One of the biggest areas of concerns has been the use of nanoparticles in sunscreen. A review this year by the Therapeutic Goods Administration found sunscreen was safe and that the particles didn't penetrate skin cells. If trends in science continue, there's no doubt nanoparticles will be used more and more.
We really need to do fairly sophisticated studies of the toxicity of nanoparticles before we use them extensively in humans.
The TGA says it will monitor scientific literature should any tangible safety concerns be identified. Sophie Scott, ABC News.
It described various dangers with biology, also nanotechnology.
Of being able to do things inside our body, inside our mind, that's going to help. What's exciting, what's terrifying about nanobots?
What's exciting is that that's a way to communicate with our neocortex, because each neocortex is pretty small and you need a small entity that can actually get in there and establish a communication channel. And that's going to really be necessary to connect our brains to AI within ourselves, because otherwise it would be hard for us to compete with it.
In a high bandwidth way.
Yeah, yeah. And that's key, actually, because a lot of the things like Neuralink are really not high bandwidth yet.
So nanobots is the way you achieve high bandwidth. How much intelligence would those nanobots have?
Yeah, they don't need a lot, just enough to basically establish a communication channel to one nanobot. So
it's primarily about communication.
Yeah.
Between external computing devices and our biological thinking machine. What worries you about nanobots? Is it similar to with the viruses?
Well, I mean, there's the gray goo challenge.
Yes.
If you had wanted to create any kind of entity and repeat itself and was able to operate in a natural environment, it could turn everything into that entity and basically destroy all biological life.
Gray goo theory is the idea that, in the future, self-replicating nanotechnology will consume anything and everything until the environment and everything in it is completely affected. This theory was proposed in 1986. It has to do with transhumanism, nanotechnology, programmable matter, and so on. It may even tie into chemtrails and Morgellons disease. But we'll get to that.
We call this method Sonogenetics. This is a tool that we can use to control neurons and other cells non-invasively. Before I get started, I want to show you what we are doing. So these are the 3 model systems that we work on in our lab. So we work with nematodes. This is nematode C. elegans. And we have been studying chemosensory behaviors now for about 10 years. And then we have built a model of fear and anxiety using the interactions between those 2 nematodes. And we've also been studying chemosensory behaviors in the zebrafish larvae and also using whole brain imaging there. And finally, most recently, we've been looking at the rodent model, particularly to test this new technology that we are developing, which I've already referred to as the Sonogenetic tool.
Hello, it is my great pleasure to share my group's latest new technology termed Sonogenetics. This is a technology that uses medical ultrasound to produce local, on-demand light emission at any place or any depth in the brain for an optogenetic brain-computer interface. In our approach, we have used mechanoluminescent nanoparticles to convert ultrasound, which has deep brain penetration, to light emission, which activates channel rhodopsin for neuromodulation. We took advantage of the endogenous circulatory system in the body. Blood vessels are not only present in deep brain for delivering nanoparticles to the ultrasound focus as a light source, but they are also abundant in superficial tissue where the vessels are shallow enough to recharge the circulating nanoparticles with the excitation light. Intravenous injection of these nanoparticles that can be charged by the 400 nanometer excitation light, which is the purple light here, when it passes through the superficial vessels in the neck and release the energy as localized blue light emission, enough to activate opsins for optogenetic neuromodulation. Forward, we envision the Sonogenetics as a non-invasive brain-computer interface that employs brain-penetrant ultrasound alone to read and write neuroactivity in the human brain. Thank you very much.
What's fascinating about these purple lights that they're calling a defect, and it's not a conspiracy or aliens or the government doing this. The news station is actually saying this.
Some thought it was some sort of discreet sign for law enforcement, the government, or aliens. No conspiracy theory behind the light.
No conspiracy, exactly. No conspiracy.
It won't let you remember.
Sorry.